great to have Dr. Peterson back. Welcome, and I won't talk long so that you can begin, because we were engaged last week, and I know we will today. Welcome.
Yeah, thank you. All right, well, good morning. Can you hear me clearly? The mic working? Okay, great. How many of you were here last Sunday? Okay, so most of you were. Good. Okay, so that helps me to know some of what we've done together. Okay. Launching into this, could we take just a moment and pray? Let's pray. Gracious Father, we pray that you would give us real discernment and wisdom, that you'd help us to understand, that you'd give us energy to focus, that you'd just anoint and bless this time with your presence. And we pray that in Jesus' name. Amen. A quick reminder as we're getting started, I'm going to be hosting Sean Hopwood at Roanoke College this Thursday. He'll be speaking at the Olin Theater, available and open to all at 7:30. Hopwood has a very interesting background that I think would be of interest to all of you. His first contact with the law was when he got caught after robbing quite a few banks. Went to federal prison. While he was in prison, there were actually a whole series of Christians who reached out to him, and there was a real transformation in his life. He ended up spending a lot of time in the law library at prison and actually successfully petitioned the U.S. Supreme Court twice and won, which most attorneys never get the opportunity to do. He finished college after he got out, after 12 years in prison, went on to earn his law degree from the University of Washington. He's now a professor of law at Georgetown University. And he's going to come and talk with us both about the transformation in his own life and also about what he thinks needs to be transformed in our system of prisons in the United States today. So it'd be very much an insider report. I think he's unique. I don't know of anybody else that's had that particular path. And so he's also very engaging. Maybe some of you saw him on 60 Minutes where they did a profile of him. So he'll be fun and interesting, and just wanted to encourage you to be aware of that. Okay, let's talk about genetics. Because I've been trained in genetics and working in this field, just about every morning I get a series of advertisements. This is one that I just got from Synthego. There are thousands of companies out there doing CRISPR now. It says, free modified synthetic RNA for CRISPR, up to 3 genome editing targets. Simply provide your target sequences. Arrives ready for transfection. This is a limited time offer. All right. So it's amazing how ubiquitous this new technology has already become. And the impact is only starting to fall into place. Just to give you an example, I pulled this information from the Wall Street Journal on Thursday, April 19th. Section B, column 3, I thought it was so interesting. This, I think, is amazing news, and it barely made it into this particular newspaper. There are about 288,000 people in the world that currently have a genetic condition called beta thalassemia. They need monthly transfusions and will likely die in their 30s, all right? So it dramatically shortens their lifespan. 3 years ago, the first cohort of 22 patients had a one-time treatment with CRISPR gene editing. 3 years later now, 15 of those 22 no longer need transfusions. They're cured. They're healthy. 7 need transfusions less often and in smaller amounts. Now, the whole process of introducing new medical treatments and cures takes time because you start with very small groups and you make sure there isn't any untoward effects. You have to figure out what's the dosage and does it affect men and women differently. There are all kinds of things we carefully do to put things in place. So it takes years to implement. But CRISPR, which was only discovered about 8 years ago, is already being applied at a level where there are people whose lives are being saved. And I think this kind of article, which was rather buried in the newspaper, you're going to see more and more. It's only going to accelerate. CRISPR is a technology that I think is going to have the kind of impact in our lives that the printing press or the computer has had. Why do I say that? Well, let me just describe very briefly what we're talking about. Bacteria can catch a cold. Bacteria can be attacked by viruses just like we can. Bacteria has developed a defense mechanism where when the foreign DNA of the virus is injected, it can spot it and cut it up into pieces. It can shred it so that it's not taken over by the virus. Well, what has just been discovered recently is how that mechanism works and that we can co-opt it for our purposes so that we can take that mechanism that naturally occurs in many kinds of bacteria and we can use it to insert or remove any genes of our choice and to change the recipe in the genes that are present, to turn genes on, to turn genes off. It's precise, it's quick, and it's cheap. And that's why it has taken the world of genetic research, just dramatically on a global basis, and why it's quickly becoming part of even high school curriculums. You can buy your own CRISPR kit for practicing CRISPR on bacteria for about $200 off the internet. So it's a little bit parallel to when personal computing started in people's garages. All right, you can do CRISPR in your garage. Now, to do CRISPR at a level of humanity is much more complex, and you start needing more education and laboratories. But the basic method is actually, again, precise and quick and inexpensive. So what does this affect? This affects anything that's alive, because everything that's alive is based on DNA. So you can just start thinking of how this will affect food, animals, pets, our own health care. It's amazing what the potential implications are. So how do we use something that's so powerful in a way to make sure that it's primarily helpful? Well, it's already being used with human embryos. And I'm just giving an example. MIT's Technology Review last June 26th, the first known attempt at creating genetically modified human embryos in the United States has been carried out by a team of researchers in Portland, Oregon. The effort, led by Shukrat Metelipov of Oregon Health and Science University, involved changing the DNA of a large number of one-cell embryos with the gene editing technique CRISPR, according to people familiar with the scientific results, and that has continued to this day. Most human embryo research is taking place in China, but there is a center in Oregon where that's being done actively. So it's not only other forms of life, it's also potentially being used with human life for treatment and for other kinds of change. Now, I have enough training in genetics. I was on grant from NIH that I could see this coming some years ago. And so my book, Genetic Turning Points, predicted 3 major waves of genetic intervention that were coming. The first wave was genetic testing. It's easier to read the human genome than it is to change it. And indeed, genetic testing is becoming quite routine standard of care. You've probably already had some sort of genetic test at some point. The first example of this being fully implemented was in 2003, the completion of the Human Genome Project, where we read one entire human genome, every base pair, all 3 billion base pairs all the way through. The next wave that we're seeing is pharmacogenomics. That's where, once you've read part of the genome, you use it to make new drugs or other treatments and services. The earliest example of this, as this was just getting started, was in 1982, the development of Humulin. A large percentage of our population, I'm sure many people in this room, have diabetes. Traditionally, if you needed to have an injection of insulin, it was porcine. It was taken from pigs, pig insulin, because that was pretty close to human insulin. Well, what we were able to do with early recombinant DNA before CRISPR, when it was much more clumsy and slow and primitive, was we found the recipe for human insulin, and we put that recipe into bacteria, commandeered their manufacturing capacity, so that you had vats of bacteria making human insulin. And so right now, diabetics in the United States who are injecting themselves with insulin are almost always using humulin, or a variation of it, which is human insulin that was made by genetically altered bacteria. Okay? So that's been quite actively and is the standard of care. That's a genetically engineered product. The third major wave that we see is genetic microsurgery or editing, and that's where you would actually go in and change genes to behave in a particular way. The first ways we could do this were called zinc fingers and talons. What's so different now, again, is that CRISPR does it quickly, accurately, and cheaply. But even as we were just starting to be able to do this, the scientists who were developing the techniques became concerned. What if somebody makes a genetic change in, say, a virus or a bacteria that's actually harmful to people, and it's now part of this living creature, and it multiplies and it gets out and becomes a danger to the general public? What if that were to happen? So in 1975, a group of the scientists who were most involved in developing this technique met at Asilomar State Park in California, and they came up with a set of guidelines that they all agreed to follow to make sure that this recombinant DNA would not be harmful. And to this day, you can find in most genetic labs charts like this that list the Asilomar guidelines. And I realize that's fine print for some of you, so I'll read it to you. Containment is essential and must match the risk. So if you're using recombinant DNA, you had to have a negative pressure lab where the air all is flowing into the lab, and any air that's leaving is going out through extremely fine filters. So if anything gets loose, it stays in the lab or gets stuck in a filter before it can get loose. Use of biological barriers to limit the spread, for example, host-specific and non-survivable. When you make a genetic change, put it into something that will die if it leaves the lab. Physical containment, good microbiological practices and training. Wash your hands. No cloning of recombinant DNAs derived from highly pathogenic organisms containing toxin genes or making biohazards that could not be contained. In other words, don't be fiddling around with an organism that's dangerous if it gets loose. So this became universally adopted among labs that did any kind of recombinant DNA work. But CRISPR adds a new dimension. Oh, let me just mention along this line, with CRISPR, if you want to know more about how it works, one of the key scientists involved in understanding this natural phenomenon and then turning it to our use was Jennifer Doudna out in San Francisco. She has a TED Talk, which is short and explains how it works. And so you can just do a quick Google search, Jennifer Doudna, CRISPR, Cas9, and you can have one of the leading scientists explain to you how this particular process works. Okay, so Asilomar 2. Genetic scientists decided that they really needed to meet again and talk about what would be proper use of this new CRISPR technology. And so December 1st to the 3rd, about 300 people met at the National Academy in Washington, D.C. The conference was sponsored by the National Academy of Medicine of the United States, the National Academy of Sciences of the United States, the National Academy of Sciences of China, and by the Royal Society of the UK. But it drew participants from all over the world who were involved in developing the technology, and then they also invited a few members of the press and a few advisors. So here's the National Academy of Sciences. Maybe you've driven by it on Constitution Ave in Washington, D.C. It's right next to the Federal Reserve Board. It's in front of the State Department, about 2 blocks from the White House. There's one of the main entrances. It's quite beautiful inside and quite ornate. It was founded by Abraham Lincoln. So Abraham Lincoln, as president, drew together leading scientists of his day and put together the funding and started the American National Academy of Sciences. Besides the beautiful interiors, it has the latest state-of-the-art conference facilities. And so this is where we met. The opening address was given by David Baltimore, Nobel Prize winner and president of the University of California. People came from all over the world. Pay no attention to that fellow in the upper right-hand corner. As with any conference, you have people who had a chance to talk with each other face-to-face, who maybe had read each other's publications, knew of each other's work, but now they have a chance to meet. And so lots of friendly greetings. That's George Daly on the left, a geneticist at Children's Hospital in Boston. Jennifer Doudna there in the center that we mentioned a moment ago, that's her. David Baltimore again. All kinds of interaction between the different academic societies. That's Rudy Janisch on the right from MIT. The fellow whose back you see there, he's the editor of the prestigious science journal from England called Nature. So quite a meeting of minds to think through the evolved issues. And in fact, the global discussion continues. This group largely met again in Paris, April 29th, then reconvened back in Washington again, July 12th, and the next official meeting is November 27th in Hong Kong. So the conversation continues. So what were we talking about? Well, I'll give you just some example of some of the key questions. Should CRISPR be used only to heal a patient presenting for treatment, or should we also treat for their future children? So someone has a condition which they're suffering from, but they're going to pass it to their kids. Should we treat their condition in such a way that it protects their children from the condition? Should gene editing only be used to cure disease, but not to enhance desired physical traits? What are desirable traits? Is it better to live longer? If we could work with CRISPR to help people live longer, should we? Would it be fair for some people to buy better genes than others can afford? Does editing someone's genes make them less of a person and more a product of our manufacture? Do we have a right or obligation to make such choices for our children and future generations? Theologically, is such playing God, or is it fulfilling a God-given calling? Who decides? So the group could look pretty pensive at times. All right, lots to think about, lots to try to figure out. And again, very much a worldwide effort. The fellow in the front left corner there, that's Nola Essen. He's the president of the Nigerian Academy of Sciences. So very much a global effort to think these things through. All right, well, there was a document that was developed as kind of a summary of a consensus of where most people were at this point. And it was released on December 3rd, 2015, at the end of the meeting. And it's interesting, it was broadcast on NPR, all the major networks, New York Times, Washington Post, London Times, I mean, on a global basis. But most people didn't notice it at all. Do you recognize that date? What else happened December 3rd, 2015? That was the San Bernardino massacre, where somebody in a public health department opened fire at the Christmas party and killed a whole bunch of people there. I mean, a terrible tragedy. And then there was all the chase and the gunfights and the rest of it. Well, that was the news. Right? So most people heard all about that and really didn't notice this, which I think is actually the global event. Okay. So the release was actually just a page or so long. It's fairly short. I'm going to walk through the whole thing with you very quickly. And it was all in white type. What's in gold is because I want to highlight it for you. So the gold is my gold. Okay? So the release begins. Scientific advances in molecular biology have produced remarkable progress in medicine. So the people involved in the development of CRISPR want it right up front and want to emphasize, remember, what we're doing is the kind of work that has protected us from polio and what has released us from smallpox. I mean, remember, we're doing research that's been crucial to our thriving and surviving in the world today. So they want that to be in the context right from the beginning. Second of all, I've highlighted in gold, the scientific community has consistently recognized its responsibility. That this is not mad scientists off in the corner creating something to destroy the world or not thinking about the implications of what they're doing. Remember, we talked about the Asilomar conference, that from the beginning of this work with genetic technology, there's been real concern that people are kept safe, that it's used properly, that we think about what's the appropriate application. So they want to say right at the beginning, remember, we're being responsible. And by the way, where was this particular event being held? Did you catch that as it went by? I mean, we even looked at the building. Washington, D.C., right? You could have had this meeting anywhere in the world. Why Washington, D.C.? Well, what's in Washington, D.C.? Okay. Number one, they're trying to show they're being responsible, they're thinking it through to head off regulation. You get lots of regulation out of Washington, D.C., right? They're trying to say we are protecting and being careful ourselves. You don't have to pass legislation that will hamstring or destroy what we're doing. So part of it is an appeal to the community in Washington that it's being carefully watched. We don't need more regulation. And number two, Washington, D.C. is one of the primary funders of this research. And so don't tell us how to do it, but keep the money coming. Okay? Do you remember, maybe some of you do, all the way back with President Nixon having a big national TV address announcing the war on cancer? Well, where did that money go for the war on cancer? Most of it went to genetics research, because cancer at its root is a genetic disease. Okay? And the most promising ways of treating cancer all go back to things we're learning about genetics. So the federal government has poured untold billions of dollars over decades into this genetics research. Now that it's looking like it's going to be extremely profitable, there's all kinds of private money going in. But it's still very important that the tax-supported dollars continue to go to this effort. And so where you're going to have a regulation meeting of the people doing it, you do it in Washington, D.C. Okay. What's new about this gene editing is that it has a much higher accuracy and efficiency than ever before possible. This is crucial, number one, for intensive basic and preclinical research, which is clearly needed. The place where CRISPR is most widely already being used is in research. If you can go in and precisely change one base pair, that is the way you spell DNA, and then see how that affects the organism, that tells you a great deal about how the organism works. So whether you're tracking people who are healthy, or whether you're looking at a disease or some sort of differently abled situation that people face, this is a tool that allows you to go in very quickly and precisely to understand how it works. So it's extremely important that it's available for research of almost every branch of the life sciences. The proposed clinical uses, where you're treating people for illnesses or disabilities, are intended to affect only the individual who receives them. This is a distinction that's called somatic therapy. Somatic just comes from the Greek word soma, which means body. And so when you do open heart surgery on one patient, you're doing a somatic therapy. You're intervening in a way that affects just that one person. And so far, they're saying, all of the clinical applications we're starting to investigate are somatic. They're not germline. They're not inheritable. We're not trying to treat future children and grandchildren. We're focusing on the patient who's been presented to us. Okay? So all treatment so far is somatic. So that beta thalassemia that I showed you about, the intention is to treat the people with beta thalassemia now. They're not trying to treat future generations or to eliminate the disease from the gene pool. They're starting with the somatic, the first patient. Germline editing, they say, raises many other important issues. There's more to think through before you make an intervention in a way that will be inherited by future generations. It would be irresponsible to proceed with any clinical use of germline editing until, and then they give some things that need to happen. In particular, be sure about safety and a broad societal consensus. As scientific knowledge advances and societal views evolve, the clinical use of germline editing should be revisited. So they're not doing germline editing now, but it should be considered for the future. Notice that's why there's that crucial word I've put on the first line in gold, until. Most of the people doing this work think that germline editing eventually will be extremely important and helpful. If you can cure the first person with Huntington's disease, why not cure them in a way that will protect their children from inheriting Huntington's disease? But at this point, they're saying there isn't sufficient societal consensus, so that shouldn't be done now. That shouldn't be done until there's societal consensus. So that means you need an ongoing forum. You need to have places where people are talking about this and thinking this through. And it should be inclusive. There was actually, I think the last two words that were added to this statement, right at the very end, were in the second to last line on your screen, 4 and 3 words in from the right, where it has this long list of people who should be part of the conversation. Providers, patients, families, people with disabilities, policymakers, regulators, research funders, and these 2 words got added right at the end, faith leaders, public industry advocates, industry representatives, and members of the general public. So there was some effort as part of the discussion that, hey, so many people in our world are influenced by their faith traditions as to their values and directions in life. Shouldn't they have a say too in this conversation? And so it, particularly through the efforts of a sociologist named John Evans, that those 2 words did get added there at the very, very end. Okay, if you want to see this all spelled out in detail, what was decided, including after further conversations for the first year, the National Academy of Sciences has published a nice thick book called Human Genome Editing, Science, Ethics, and Governance. It's available in print for $68, or you can download it for free. They want people to have access to it and see it for themselves. So if you want to see what the National Academy of Sciences, as part of a global effort, so far has agreed is appropriate use, that's where you can download it for free, or you can purchase a print copy. If you want to think about it from specifically a Christian perspective, that's what my book, Changing Human Nature, tries to do. The whole book is thinking through, now that CRISPR is available and in place, what is the appropriate response and use from a Christian perspective. So I can vouch for this one. I think this one makes sense. Okay. All right, well, what we could start to talk about next is what are some of the ways people have thought about proper and improper use. But let's take a moment to make sure that we're all on the same page. What would you like to ask about or challenge or consider so far? I know that we've covered a lot of material already. Yes, sir. Yeah, I don't know if you could hear in the back, the question was raised, It's so easy to travel these days. Why couldn't somebody just go to a different country if they have the money and do something unethical with this technology? It's a very genuine concern, and that's why the very first meeting and what has followed were intentionally joint meetings of the Chinese Academy as well as the Royal Academy as well as the U.S. Academy. There's been a real effort among people in the field who know how this can be done to try to build a consensus, an agreement about what's appropriate and what's not, and try to keep everybody together. Within those communities, it's extremely important that you have a good reputation because you're sharing information and you're sharing biologics back and forth. And so if you get a reputation for being off the reservation, for abusing or not properly using these things, you would quickly be isolated, and not only would you not win a Nobel Prize, you're going to have trouble just getting the reagents that you need for your next experiment. So there is a very strong feedback loop that keeps people together that way. People are concerned about their reputation and about their funding and about their access. Could you have a country like a North Korea that's a pariah anyway, doing whatever they want? Could you have them going off their own way? Yes, and this is a real concern. The most scary slide I saw at this first meeting was from a professor at Stanford who specializes in defense issues, and he showed the head of the physics department at the largest city that was taken over by ISIS, back when ISIS had some territory. And this was a physics professor who had been beheaded by ISIS because he refused to help them make a bomb. And then he simply said, there was also a biology department at the same university where we're not aware of anybody being beheaded. So what is the risk that a group like ISIS can get a hold of this technology and use it badly? It's a very real risk. What's our number one line of defense? It would be the Centers for Disease Control in Atlanta, and then also Fort Derrick in Maryland. So the United States does have organizations who try to stay on top of the latest technology and are thinking in terms of how do we defend ourselves if somebody uses this, say, to make a infectious pathogen that would be dangerous. So that is a real concern and very appropriate that you raise it, and there are people who are thinking about it and trying to make sure that we're ready to move quickly when there is some sort of abuse. Yeah. Every power creates good and bad possibilities, doesn't it? Yeah, very, very thoughtful to raise that. Yes, sir.
Could you expand on what you were just saying a little bit more in terms of optimal or most glorious benefits of CRISPR in terms of theory, and then worst case scenario, disastrous things that could happen, expanding on what you were just talking about, so we can get a better sense of possibilities here.
Well, just some more quick examples, and I'm sure I haven't thought of the best uses and I haven't thought of the worst uses. Just think of any new technology. When we started putting personal computing power into individual homes and then eventually into our very telephones, nobody was really talking 30 years ago about what would be the effect of children being raised on Facebook and constantly having to defend and explain who they are in a public forum. There are just all kinds of effects that I'm sure I haven't thought of and I'm not aware of. What are some possible negative effects? Well, one would be indeed this idea of it being used as a weapon, weaponizing. Positive effects, well, for example, many of the people in the world who are alive today are alive because of the agricultural revolution that has come through using genetic technology to improve the productivity of our farmers. To add vitamins to rice, like to add vitamin A to golden rice. There are countless millions of people in the world today who are able to see because their body was able to get vitamin A from their rice that would have gone blind 20 years ago. Just the list goes on and on. We're facing Zika, the virus being passed by mosquitoes. We have millions of people who are dying from malaria every year. We have the genetic capability now with CRISPR to make the 1 out of 3,600 species of mosquitoes that carry malaria immune to malaria. So if you could genetically alter those mosquitoes so they'd no longer get sick with malaria, they'd no longer pass it to people, and we could eliminate malaria. It's very much within the technique. It's just a question of, are we confident enough that we're willing to release this into the general environment? And so there are all kinds of studies going on with that right now. So the ability to affect disease is amazing. The ability to eliminate genetic disease. There was a pediatrician here last week. Is our pediatrician still here? Okay. What percentage of your patients as a pediatrician that are hospitalized in some part due to a genetic condition? And is that because of the nature of your particular practice? Yeah, I mean, what I'm generally told is among children who are hospitalized, a genetic component is a major part of their condition is actually quite high. Yeah. So there are all kinds of ways and implications that this can be extremely helpful and useful. But basically, again, it's the potential to intervene in anything that's alive. So just think of everything that's alive. If there's anything untoward, there's the potential to intervene. So incredible upside. Yes, like any new powerful technology, there are ways it can be abused too. Yes, sir. Right. Let's see. Most diseases have a genetic component, but are not solely determined by genes. So, for example, if you have identical twins, you have two people raised in the same family and environment with the same genes, and one of them comes down with juvenile onset diabetes, what's the chance that their identical twin will come down with juvenile onset diabetes? It's about 50%. Now that's really interesting, isn't it? That tells you that's so much higher than the general population. That tells you that the genes make a tremendous difference. But if it was just genetic, the identical twin would have a 100% chance of coming down with the disease. So that tells you the genes play a major role, but they're not determinative just by themselves. Okay, so most diseases that we face have a genetic component, but they're not determined just by genes. An exception to that would be, for example, Huntington's, which is just straight out a genetic disease. If it's a straight out just genetic disease, and you can go in and correct the genetic issue, then it should be possible to cure. Now, life is always complex and messy. I gave you the example of beta thalassemia, which is one of the earliest cures to come down the line. For the 22 people who had the first treatment, 15 of them are healthy, and there are 7 of them that are much better but still need some treatment. So what's going on there? We just don't know exactly at this point. So there are some conditions that we face that are purely genetic, that you can have a purely genetic resolution. Most diseases, it's a component. And then there are things we can do genetically to help your body deal with something. For example, vaccines are where we're teaching your body to deal with a particular threat. We're hyping up the immune system to handle something it didn't know how to handle before. Well, we know that some people have a higher natural resistance to cancer. So if we can find that recipe, if you give that recipe to somebody else, then they would have a higher natural resistance to cancer. So, I mean, the implications are it will vary in its intensity of effect, but the different kinds of ways it could be used, we haven't even thought of all the possibilities. Yeah. So maybe that gives some idea to start to think about.
At these conferences, there's a general agreement as to the responsible way to proceed.
At least so far, yes.
So far.
Okay.
But it's a and you talked about how to deal with bad actor nations. Mm-hmm. What about, I assume that all the people doing all the work are not homogeneous, and so there are some bad actors individually. Has there been any thought given to how to proceed systematically with that?
Well, in the sense that, for example, if you want to get funding,
I understand from the research, but
right,
I'm talking about
or just applying it other
applying down the road. Hey, go on the dark web and your children will be an NFL player or something like that.
Okay, yeah. For complex behaviors like being an NFL player, we're not there yet.
No, I understand.
But could you have a black market for something that you believe is an enhancement for your child that hasn't been widely approved or used yet? Yes. And in fact, I think probably the way this is going to go is that most of the initial experiments and efforts to see what would be an enhancement for a child will be tried out on rich kids. So there'll be the same parents who would spend a heck of a lot of money to give their kid the best college education might very well want to buy other advantages besides piano lessons for their children. And so probably our initial test subjects for enhancements will indeed be rich kids, wealthy families, where they'll be trying to purchase an advantage for their children. Once it's shown that it's safe and effective, the price will plummet, and there'll be a lot of pressure on government to provide it for everybody. And the economic issue would be the government will want to provide it if it really is an advantage, if it makes the citizen more productive to pay higher taxes in the future or to be less demanding on the government for care. The economic pressure will be on the government to provide it in terms of the way it's likely to unfold in the future. The biggest issue will be for people who don't want to have it for themselves or their children. And then we have the same situation as we do with vaccines, where we want to require it for the good of the individual, the child, and the larger community. But what if the parents don't want it for their child, and how do we handle that? That tug and pull, I think we'll have that kind of tug and pull in the future in regard to genetic enhancements. And there's a lot more to unpack there, but that's a good thing to put on the table that we should talk about, yeah. Let's see, cloning is where you try to duplicate one particular person's genes in the next generation. It's kind of an intentional, delayed, identical twin. Would it ever be appropriate to intentionally create delayed, identical twins? Well, the first thing to remember is that any person that starts out that way, like an identical twin, would be a full, independent person and would make their own choices and become a unique individual. So they wouldn't be disposable or just an extra copy. They'd be a fellow human being. Can we think of scenarios where that would be appropriate? If you want to talk about that, we could. That's a whole other discussion and an interesting one. Okay.
We have about 10 minutes. So, Dr. Peterson, do you want to do this, or do you want to press forward a little bit before?
Well, I think as long as we're finding this useful, I think people have questions.
Fine.
It's okay with me. We have next Sunday to lay out a Christian perspective on all this.
I just want to make a personal observation, and maybe you react to it, but ask a question too. It seems to me that there's something inherently incongruent in our overall behavior. You know, Hippocrates didn't want to do any harm, and from the time science has begun, at least the sciences that we're talking about, if we conceptualize it as a fairly broad scenario, the goal has been always to get to the fine point where health could be managed or controlled, or life could be improved or elongated or something. And the closer we get to the ability to do that, the less sure we are that we should, and that's the incongruity. And the question is, don't we at last have to simply take some calculated risk because we can never anticipate all the possible benefits or all the potential liabilities?
Yeah, I don't think it's possible to predict every possible result of a new intervention. And that's why traditionally in medical care, we're very careful. We start with small groups and we test for toxicity. Is this new potential pharmaceutical have bad side effects? Does it cause it harm? Once we're confident that it seems to be safe, then we do very careful testing for dosage, and we just very gradually expand the circle out wider and wider. So we try to avoid massive mistakes, and sometimes bad mistakes still get in there. If something has a bad effect on only 1 out of 100,000 people, and you test it on 80,000, you don't know that it's bad for the 1 in 100,000 until it's in the general public and 1 million people are taking it. So, I mean, that's why things are always kind of gradually released and you're looking as you go. But can you be totally risk-free? That's probably just not possible. At least not, you can't do something new without taking some risk. But we try to be as thoughtful and careful as we can ahead of time. Let me go ahead and outline some directions that I think we should consider. And next Sunday we'll have a chance to go through much more detail. I'd like to talk about things that Jesus taught. I'd like to talk about things Christians have thought about this, and try to work through it much more specifically as a Christian perspective. One of the distinctions we saw was this distinction between somatic and germline. Right now, it's just used to affect the first body, not to affect their children. This can be a useful distinction about what you're trying to do, but it actually gets murky very quickly. For example, the first genetic test that became standard of care that we do for all babies that are born, we don't ask the parents, we just automatically do it, is a blood spot for PKU. And why do we do that? We do that because we don't have a cure for PKU, but if we change your diet, if we remove phenylketonuria, a particular chemical from your food, even if you have the condition, your body doesn't know how to break it up. If you don't eat it, it won't accumulate and arrest your mental development. So we can't cure that disease, but if we know you have it as a baby, we can put you on a special diet for the rest of your life. It doesn't include pizza or hamburgers. It's a bummer, all right? It's kind of bland. But if you're on that diet, you can lead a normal life. Okay? So it's not a cure, but you can avoid the effects of the disease. So that's done on every baby born in Virginia. There's a test for PKU to see if they need to be put on that special diet. We've been doing that now for over 30 years. And that we have discovered a germline effect, and you can probably think what it is. You put it together, what's happening? That the incidence of PKU is going up dramatically. Why? They're having children. It used to be, if you had the condition of PKU, your mental development would be arrested at the infantile level. I mean, devastating mental damage. And you'd never grow up to have children. But now we have people who have been protected from PKU, who grow up, and like everybody else, they get married and have kids and pass on their PKU to their children. Right? So it's become a germline effect. And I'm just giving that as a very concrete example, that the distinction between somatic and germline gets fuzzy very quickly. Also, there are many genetic conditions where if you intervene early enough to make a difference, you intervene early enough to affect the precursor cells that create eggs and sperm, and then that affects the next generation. So somatic and germline is a current key distinction, but actually in practice we're finding it's a tough one to make. Some people have said we should use genetic intervention to cure, but not to enhance. But this then requires you to differentiate between curing and enhancing. In a way, you can say that any cure is enhancing the person's life. So what is the difference between cure and enhancement? And there's a whole discussion about that. Some people say we should welcome every child as they are when they're born, because if you intervene, you're making them into a product of manufacture. And we're running out of time. I'll just say, if a child's born with a cleft palate, and we do surgery to bring their palate and lip together so that they can eat and speak, we've dramatically intervened surgically right at the beginning. Does that mean we now think of them as an object rather than as a person? Maybe we did it because we think of them as a person and really care about them. Does it make them any less because we intervened? I would argue not. Well, there are a lot of distinctions that have been proposed. Next time we meet, I want to talk specifically from a very biblical and theological approach about how we might think about these distinctions. Since we did promise we're going to do this from a Christian perspective, I've been laying the groundwork, explaining where we are and what's going on. All right, so we've run out of time.
Yeah, thanks,
We'll
Dr.
reconvene in
Peterson.
a week.
That's a great lead-in to force you to come back next week to talk about Jesus just a little bit. So, thank you, Dr. Peterson. That's obviously very fascinated by all this, and we look forward to kind of summing it up next week. So if you've been to worship, thank you. We'll see you next Sunday. If you are out to head there, you can go now and get a really good seat still. So we'll see you in worship.